So now that you've gone through a few examples of inserting a sequence into an organism to get a desired trait, we wanted to explain to you one of the projects that we've been working with. I'm a part of the Butanol group, and what we're trying to do is make a moss called Fiscomitrella synthetically produce butanol. We're trying to do this because right now, to get butanol, we will grow entire fields of crops and then raise them all down, collect all that biomass, and then break it down just to get a portion of that crop to turn it into butanol. The idea behind this is if we could get the plant to directly produce the butanol, you could just extract it, and then you wouldn't have to go through that cycle. We're nowhere near that step right now. This is just a proof of concept. But what we're doing is taking an enzyme chain that was used by the University of California to make butanol within cyanobacteria, and we're trying to take that same sequence and put it within Fiscomitrella. So you did a similar thing. Once you try to do something a bit more complicated than that, it gets kind of overwhelming. As you can tell, it's much longer sequences, and we're trying to put multiple protein coding sequences within each promoter terminator boundary. We're doing that by using two-way tags, which is something that has only been used successfully 2 or 3 times before. So we're really crossing our fingers that it works. It easily could not, though. So if you insert our first sequence, it will get you to, it will take compounds that are naturally within the moss, combine them, adjust them, and to reach this intermediary compound, which, if you add our second enzyme sequence, will adjust it and turn it into butanol. We came up with a second goal in this project when we realized that that intermediary compound that this first enzyme chain was going to produce, all you have to do is add one more enzyme to combine 2 of those compounds, and it'll make a biodegradable plastic, which we thought would be another industrially relevant, useful resource to produce. So if you add, instead of this second genome sequence and add this third genome sequence, you'll get a biodegradable plastic. So there are many different applications for synthetic biology, and this is one of them, and we hope it works.
Any questions in general? Well, thank you guys for coming today. We really enjoyed it. You guys did an awesome, awesome job. We hope you guys had a little bit of fun, and enjoy the rest of your time at Penn State, and have a really good week. Thank you.
Before we go, does anyone have any questions for the iGEM group, just in general? Matt?
So what else is the two-way tags used for, and how do they work?
Okay, I will answer how they work first, because I actually know that answer. Instead of having each promoter make the protein that you want and then stop, it makes this long polypeptide chain, just this huge protein that doesn't actually do anything, and then the two-way tag is just this series within that protein that destroys itself. So you make this giant protein that doesn't do anything, and then the little two-way tags within fall apart, so then they all hopefully will spread away from each other. You'll have all the different proteins you need. The reason we didn't just do protein stop, promoter stop, promoter stop, promoter stop, is because if a plant has too many promoters within one region, it'll be like, well, this never happens in nature, and it will shut down the entire region. So this is a way of sneaking around that. Kevin, do you know what two-way tags have been used for in the past?
They originally were derived from the foot-and-mouth disease virus. So viruses have very small genomes, they try to be very efficient. So they figured out how to make a two-way sequence. They've been used, as far as I know, they've been used to express a couple of proteins in rice. They were also used in a paper to express the 4 cytokines for making induced pluripotent stem cells and training all those together in a single sequence successfully.
That's another thing that I find really cool about synthetic biology. The two-way tag was found from something that was in the foot-mouth virus. Some of our proteins we took from orangutans. One of them I think is from a platypus, and we're putting it in a moss. So it's, you can really find these proteins anywhere you want because all organisms, since they're all based on DNA, have a certain amount of similarity. So you can just pick and choose from wherever you want, even platypuses. Platypod. Any other questions? Okay.